So good afternoon, everyone. I would like to welcome you to the second AGePOP Open Research webinar with the title Gastric Emptying and Luminal Contents in Advanced Age Population, Current Knowledge and Missing Data. Before we start, I would like to present a brief overview of our consortium. AGePOP is the acronym of this Marie Curie action and stands for Drug Absorption in Geriatric Patients and Older People, a training network innovating drug development for the advanced age population. The objective of AGePOP is to generate for the first time information on the actual dosage form intake conditions for older people and geriatric patients and on physiological characteristics of their gastrointestinal tract relevant to oral drug absorption. AGePOP team consists of 3 academic institutions with research excellence in oral drug absorption and development, KU Leuven, University of Greifswald, and National and Kapodistrian University of Athens, and the 3 leading pharmaceutical companies, Bayer from Germany, Roche in Switzerland, and Merck in Germany. In addition, 8 partner organizations from 5 countries from 3 different sectors complete the AGePOP consortium. From the clinical sector, KU Leuven universities participate to this consortium, University Greifswald Hospital, and also the Red Cross Hospital of Athens. From the leading pharmaceutical companies, we have together with us Janssen in Belgium, AstraZeneca in UK, UCB in Belgium, Novartis in Switzerland, and AbbVie in Germany. And also we have one SME with expertise in European trainings and collaborate with us and this development. Then 11 ESRs from 8 countries are on board, and our aim is to provide them with the opportunity to become the first trained experts on oral drug delivery of drug products to older people and geriatric patients in Europe. 2 of our ESRs are going to present today their research in this second AGePOP webinar. AGePOP is funded from EU Horizon 2020 with 2.8 million euros and is a Marie Curie action. It's a European training program. We started on January 2021, and for further information and updates, you are welcome to visit our website and follow us in LinkedIn and Twitter. And now it's my pleasure to introduce you our moderator for today, Professor Christos Repas. He's professor in pharmaceutics at the Department of Pharmacy of the National Kapodistrian University of Athens. He's also the work package 2 leader in AGePOP and supervisors of a few of our ESRs. So, Christos, I would like to give the stage to you.
Yeah, thank you, Maria. I would also like to welcome you all at the second AGePOP webinar on gastric emptying and luminal contents in the advanced age population. The subject will be discussed by 2 AGePOP early stage researchers, Theodora Tzakri in the beginning and Ekaterina Bocharova after Theodora. I would like to encourage participants to submit questions using the chat tool at any time during the presentations. At the end of the 2 presentations, I will address the questions verbally to Theodora and Ekaterina. So Theodora Tzakri, an early stage researcher who is working at the university, based at the University of Greifswald in Germany, will start this webinar. Theodora, please, if you can share your screen.
Thank you, Professor Repas. Welcome from my side. I'm Theodora and I'm a PhD student in the University of Greifswald. And today, together with Ekaterina, we're going to present you the second online AGePOP webinar on gastric emptying and luminal contents in advanced AIDS populations, current knowledge and missing data. First, I'm going to present you an outline of our today's webinar. First, I will show you a short introduction on our topic and then continue on some details about gastric emptying and oral drug absorption in the advanced AIDS. And then Ekaterina will continue with her project on luminal content characteristics and advanced AIDS, and our conclusion of our webinar. The advanced AIDS population, meaning people aged 65 years and older, is a really growing population nowadays. As you can see on the map, we have here Europe. These are the statistics from Eurostat. There are some regions in Europe that the advanced AIDS population is more than 25%. Actually, in 2022 in the EU, we had a 21.1% percentage of older people in the total population. And this number is expected to grow even more. And until 2050, it is estimated that in the EU, there will be 150 million inhabitants of older people, which correspond to more than 1/4 of the total world population. This special population, the advanced AIDS population, has some characteristics. First of all, the oral route drug administration is the most preferred, and this is because of patient compliance, ease of administration, and non-invasiveness. Some characteristics of this population are, among others, multimorbidity, increased pharmacotherapy, frailty, a high prevalence of gastrointestinal surgery, neurodegenerative diseases, and a broad heterogeneity. The point is that nowadays, in clinical trials of new drug entities, data from younger adults are being used, and also young volunteers participate in these studies. However, older people are the main consumer of formulations. For oral drug absorption, there are certain physiological determinants which play an important role, such as the gastric emptying, the luminal contents, and the intestinal permeability. On today's webinar, we're going to present you about gastric emptying and luminal contents. To identify these characteristics, me and all the other PhD students from AGePOP, first we collected all the current knowledge and information regarding oral drug absorption in older people. Then we identified the missing data, and currently we're working on filling these research gaps while we're doing our clinical studies on older people. And the goal is to enrich in existing biopharmaceutical in vitro and in silico tools for a patient-centric drug development for older patients. So to continue, let's see some more details about gastric emptying and oral drug absorption in the advanced AIDS. Firstly, let's consider why is gastric emptying important for oral drug absorption. After we swallow a formulation, the first organ from the gastrointestinal tract that comes into contact with this formulation is our stomach. The formulation has some specific characteristics, and the drugs needs to be liberated from the formulation, then mixed with the gastric contents, and after being emptied from the stomach to the intestines, it can be absorbed and have some pharmacological results. The state of the stomach, meaning if we have eaten or not, plays an important factor for oral drug absorption. In the clinical trial conditions, we have two prandial states. We have the fasted state, which means that the person has an empty stomach and has fasted overnight. In this case, the volunteer takes the formulation together with a glass of water. And we also have the fed state, which is after the intake of the FDA high-fat, high-caloric standard meal or a similar meal to it. And then the person takes the formulation together with water. But in this case, we have a full stomach. So the prandial state has an important plays an important a role for all drug absorption because we may have food effects, meaning we may have a negative or positive food effect. Also, the rate of gastric emptying is really important since a faster gastric emptying may result in a higher onset of pharmacological effect. Or if we have a delayed gastric emptying, then we may have a delayed onset of action. Also, in the fed state, we have the appearance of the Magenstrasse or the stomach road, which is actually a really rapid emptying of the ingested water in the fed state conditions. This is of particular importance, especially for high soluble and high permeable drugs with a rapid dissolution. However, let's see in more detail some characteristics of the physiology of the aging stomach. In the aging stomach, we have a decreased homeostatic repair mechanism, gastrointestinal chronic diseases, other systemic chronic diseases such as diabetes and chronic renal failure, changes in the migratory motor complex, reduced mucosal protective mechanisms, and a decreased gastric blood flow. Also, we have a high intake of medications in the older people, which affect the gastric motility and gastric pH. In my research project, I'm mainly focused on the gastric emptying of liquids because in the clinical trial conditions in both fasted and fed state, the oral formulations are being ingested together with water. I did a literature research to find previous studies, and there I find only a small amount of studies that they investigated the gastric emptying of liquids in both young and older people. And actually only one study investigated the gastric emptying of water. And here they found that the gastric health emptying time of older men was slower in comparison to young people. However, it is important to mention that with these studies, we can see that they used various protocols, different methods, and they didn't always consider frailty. Consequently, we have different results and conflicting results, meaning we cannot make a solid statement and say, yes, gastric emptying was slower or didn't have any change at all. To measure gastric emptying, there are many methods that are being used until now, such as magnetic resonance imaging, scintigraphy, breath tests, and intubation and aspiration of gastric contents, and the paracetamol absorption technique. But to measure gastric emptying in older people, we need a patient-friendly, simple, non-invasive, and easily monitored method. The salivary tracer technique, previously developed from our working group, is a non-invasive method that can be used to measure gastric emptying in older people. This technique uses caffeine as a salivary marker, since caffeine can be easily absorbed from the small intestine, resulting in a good correlation between blood plasma concentration and saliva concentration. The method is non-invasive and is already validated with magnetic resonance imaging. The volunteer, after ingesting caffeine, can give saliva samples and then in specific time points, and then by measuring the caffeine concentrations, we can determine the gastric emptying rate. However, it's important to have a vehicle so that we can deliver caffeine to the stomach. Firstly, the ice capsule was used to deliver caffeine to the stomach. However, this formulation had some limitations and it was challenging to use it in older people. First of all, it was melting really fast in room temperature and also in the oral cavity, resulting in the failure of the method. Also, it was difficult to use this formulation in different clinical trial settings like hospitals, so we needed to find another alternative. And last but not least, this formulation had a quite big size of 15 millimeters diameter total, and this was also challenging to use it to older people who may have swallowing difficulties. So we came up with the idea of creating a new formulation that could be easier to use on older people. So we developed a tablet that contains caffeine in the middle. And this tablet has many advantages. First of all, it has a relatively small size of 9mm diameter and is really easily to swallow, and it can stay stable in room temperature. And we also did a stability study, and we saw that up to 6 months, the tablet remains stable. Then we validated this new formulation in comparison with the ice capsule in a clinical study, a crossover study under fasted and fed state conditions in young volunteers. And as you can see our results in both fasted and fed state, we can see that the two formulations behave similar to each other, and we can detect gastric water emptying in both conditions. These results we have already published in our open access article on pharmaceutics a few months ago. And to continue and to fill in the research gap on gastric emptying in older volunteers, I'm running right now a clinical study using the new formulation, the tablet, and under fasted and fed state conditions in a similar setting like the young study, the young volunteer study. And in this case, we have the ingestion of the light breakfast because ingesting FDA breakfast in such an advanced age could be challenging for many of our volunteers. And this study is still being investigated, and we're planning to publish these results this year. So after collecting all my data, I want to close the gaps of gastric emptying in older people and investigate any changes in relation to age, pharmacotherapy, or prandial state. Thank you very much. Now we continue with Ekaterina.
Thank you, Theodora. Thank you, Professor Repes. Okay, and now I will continue with talking about luminal contents characteristics and advanced stage. And first of all, I would like to explain or remind you why studying luminal characteristics is important for oral drug absorption. Well, the first parameter to consider is pH, which affects the ionization of molecules and dissolution or stability of drugs in certain drug delivery systems. Another parameter is buffer capacity, which is defined by the ability of the solution to resist pH changes, and it affects the degree of ionization of drug molecules and drug solubility in the bulk contents. Another important parameter is osmolality, which affects interluminal volumes and indirectly reflects ionic strength, which is particularly important for some for drug dissolution and drug release from certain drug products. Another parameter of interest is biliary lipids content in luminal contents, which can affect the solubility and the dissolution rate of some drugs, and especially lipophilic drugs. And last but not least are the digestive enzymes that are present in luminal contents in some regions, including lipases and pepsin. And lipases, both gastric lipase and pancreatic lipase, can impact performance of lipase-based formulations and can contribute to the lowering of surface tension. And pepsin, which is mostly present in gastric luminal contents, can impact performance of peptides and also contributes to the lowering of surface tension in the gastric contents. So now we will talk about the published data in stomach and for certain characteristics such as volume, acid output, pepsin output, and gastric lipase activity. And now the first characteristic is the volume of contents, which have been studied in the fasted state. And it has been shown that the mean resting volume of gastric fluids is decreased in older people. However, the differences were not statistically significant. The next parameter is acid output, which reflects the rate of secretion of acid into the gastric lumen. And basal acid output, which reflects acid output in the fasted state, and peak acid output, which mimics fat state, has been shown to decrease with advanced stage. However, after employing multivariate analysis, it's been shown that age as itself does not have an impact on these parameters, and the decrease was due to the net result of confounding factors, including smoking, H. pylori infection, and atrophic gastritis. Related to acid output is the gastric pH, and here we have data from 2 different studies. And in one study showed that in the fasted state, older adults had significantly lower pH in comparison with young adults, and there were no differences in the fat state. And in the second study, it's been shown that there were no differences between the age groups in gastric pH, and there was a trend for a slight increase in pH values in older adults. It should be noted that in the first study, smokers were included as participants, and water was allowed ad libitum. And in the second study, smokers were not included, and no water administration was allowed 2 hours prior to the investigation. The next parameter is pepsin output, which reflects the secretion of pepsin into the gastric lumen. Basal pepsin output reflects the pepsin output in the fasted state, and pentagastrin simulated pepsin output reflects the fat state. And it's also been shown to, both of the parameters were shown to decrease with advanced stage. And here, unlike with acid output, multivariate analysis showed that advanced stage does have an impact on pepsin output, along with the confounding factors, smoking, H. pylori infection, and atrophic gastritis. And the final parameter for gastric contents is activity of gastric lipase, which has been investigated in fundic mucosa and not in luminal contents. And it's been shown to decrease in advanced stage. However, it should be noted that in this study, prandial state was not specified. And now moving along gastrointestinal tract, we're going to the intestinal contents. And here we will talk about volume, pH, buffer capacity, pancreatic lipase activity, osmolality, bile salts, and cholesterol. So the first parameter is volume of contents in the lower intestine, where we have the data available from 2 regions, distal ileum and proximal colon. And in distal ileum in the fasted state, the volume of contents was significantly decreased in older people, but was not different there was no differences in the fat state. In proximal colon, in both prandial states, older people showed a significant decrease in volume of contents. Hence, volume of contents of distal ileum and proximal colon seems to be lower in older people. The next parameter is pH, and the data are available from 2 studies that aimed to investigate the impact of H on duodenal pH. In one study, it's been reported that older adults had significantly higher pH in duodenum in the fasted state, as well as in the fat state. However, another study in the fasted state showed that there were no differences in pH in duodenum in the fasted state. And we continue with pH in lower intestine, in distal ileum and proximal colon. And while in the fasted state, there were no differences in pH between young and older people. In distal ileum, in fed state, the pH was significantly decreased in older people. And likewise, in proximal colon, in both fasted and fed state, pH of contents was significantly lower in older adults. So pH of contents of distal ileum and proximal colon seems to be lower in older adults. Now moving on to buffer capacity. For distal ileum, the data are available in the fed state, and both of these two measurements were independently investigated. They were higher than in the young adults. And in proximal colon, buffer capacity in both prandial states was significantly higher in older adults in comparison with young adults. So buffer capacity of contents of proximal colon seems to be higher in older adults. The next parameter is osmolality, for which we have the data from 3 regions: duodenum, distal ileum, and proximal colon. And in duodenum, since it seems that there are no differences in between the age groups in the fasted state. In distal ileum, as well as in proximal colon in fasted state, osmolality is significantly increased in older people in comparison with young. And in fed state, neither in distal ileum nor in proximal colon, there were no differences between the age groups. So osmolality of contents of distal ileum and proximal colon seems to be higher in older adults in the fasted state. The next parameter is pancreatic lipase activity, which have been studied in duodenal contents after hormonal stimulus in order to trigger the secretion of pancreatic juices into duodenum. And pancreatic lipase concentrations seem to highly vary, but at the same time, pancreatic lipase concentration linearly decreased with age, and this decrease was statistically significant. The next parameter is total concentration of bile acids, and while in distal ileum, in neither of prandial states, there were no differences between age groups. In proximal colon, in fasted state, older adults showed higher concentration of total bile salts in comparison with young people, and in the fed state, lower content of total bile salts. So total bile acid content in proximal colon seems to be altered in older people. And the final characteristic for intestinal contents is cholesterol concentration. And here, neither in distal ileum nor in proximal colon, in neither of the prandial states, there were any differences between young and older subjects. So, however, there was a trend to increase cholesterol concentrations in older people. And here is a summary of published data on the impact of advanced age on characteristics of luminal contents. And for many parameters in many regions, the data are completely lacking from the literature, while other parameters were investigated in one study. And if at least 2 studies investigated the impact of age on a characteristic, the results were inconclusive and conflicting, probably due to disregard of confounding factors and limited amount of participants. And this is where our project comes in, which aims to close this gap and fill in the gap. In our work group, we studied the impact of aging as well as mild mild cognitive impairment, on physicochemical characteristics, luminal contents in stomach and duodenum in the fasted state, and we took into account the limitations of previous studies and considered potential confounding factors such as smoking and comorbidities, for example, diabetes, as well as co-medication. And we plan to publish the results of our studies this year. So if you're interested, please stay tuned. And to conclude, understanding the physiological alterations associated with aging is vital for drug product performance in older adults and is necessary for improvement of predictive in vitro tools and in silico models, and employment of such tools will allow assessment of absorption without in vivo studies in humans and animals, and identifying the rate limiting steps will ensure that early on the correct formulation strategies can be employed, which in the end will result in improved absorption and better patient outcomes. If you're interested more and you would like to learn more details, please check out our review that has been recently published in European Journal of Pharmaceutical Sciences in the special issue of dedicated to AGePOP. It is an open access article, so feel free to read. And on this note, I would like to thank you all and also acknowledge our supervisors. And in case you have any questions or later you come up with some questions, do not hesitate to contact either Theodora or myself. And with that, I would like to thank you for your attention, and we will be happy to answer any of your questions.
Thank you, Ekaterina. So, yeah, I mean, there are a couple of questions already received. One question that goes to Theodora and relates to the caffeine dose that the volunteers are administered. How much caffeine is the single dose?
Yeah, it's approximately 25 milligrams of caffeine, and this amount of caffeine is quite low, so we ensure that we have no pharmacological effect. And we use caffeine completely as a marker.
Yeah, thank you. Yeah, another question also for you, Theodora. It's a general one. Could you briefly say on the importance to the type of food or to the amount of food on gastric emptying? Very, very, very briefly. Yeah. I mean, it's the amount of calories that affect the gastric emptying. It's the type of calories.
For a solid gastric emptying, so for food is the caloric density. It's approximately 2 to 4 kilocalories per minute. But for water is different, and of course we have the effect of the Magenstrasse, and what I'm actually studying on my research project is the emptying of water.
Thank you. Any information so far on the potential differences on gastric motility and strength of contractions between old adults and young adults?
From my study, I still have no data, but from the literature, as I mentioned before, there were some information about the physiological changes. Of course, a relaxation of the gastric muscles, yes, in older people.
Thank you. There is one question for Ekaterina. Ekaterina, would you say that the smaller volumes in the colon of older adults could be related to the decreased food intake in advanced age?
Well, in general, probably, but in terms of the results from the studies that I presented, both young and older volunteers consumed the same meal, so the conditions were completely the same, and they followed the same protocol. So in this case, I would say no.
Mm-hmm. Well, these are, let me see, yeah, there is one more question just in. Well, yeah, I think it goes again to Theodora. In regard to the Magenstrasse, to the stomach road, is the importance of this path linked to the type of meal first and to the formulation second?
So from previous studies that were done here in the University of Greifswald with MRI, they found that the Magenstrasse appears regardless of the type of meal. Also, there is an interesting study from Dr. Grimm that you can see online. And formulation, of course, the Magenstrasse affects formulations that from drugs that are high soluble and high permeable, and either they have really fast disintegration, like immediate release formulation, rapid disintegrating, or oral solutions, of course. Yes, is the answer for this question.
But it has to be connected with the density of the meal. I mean, it refers to the liquid or to the water, right? Phase. Yeah.
The water phase is the Magenstrasse, yes.
Thank you. Yeah, a general question again. The participated volunteers or patients or the number of data come from a limited number of individuals. Would you think, both of you, we could generalize information at this stage? We can start with Theodora, and then we can go to Ekaterina.
I would say that no, we cannot generalize. First of all, because from previous studies, we can see that either the characteristics of the volunteers was not correct, was not properly documented, and we have many missing data. And also, it's important, I think, to make some subgroups, and at least at this stage now, we cannot say that, yeah, we have these previously published data that correspond to the whole older people population. No, I don't think that is representative.
Rina?
Yes, I would like to add to that, that as Theodora mentioned in the beginning of her presentation, that advanced stage population is highly heterogeneous. Older people do suffer from different diseases, they do take different drugs, and everything has to be taken into account. Regarding the luminal contents, for example, it's very important to take into account confounding factors. So the participants selected to participate in the studies have to be very carefully considered, so that the results that we get are not contaminated, but we get the results just because of the age effect and not something else. Because, for example, older people suffer from gastritis more, or they have higher rates of H. pylori infection, and we have to take all these parameters into account.
Thank you. I guess there are no other questions to ask. Yeah, at this stage, therefore, I would like to thank Theodora and Ekaterina for the presentation and the discussion. I think that it was interesting.
Thank you. I would like to thank you, all the attendees for their interest in our consortium. Thank Dora and Ekaterina for the nice presentation, and you, Professor Epos, for the moderating of this webinar. And I'm happy to announce you the third AGePOP Open Research webinar. The topic will be physiologically based approaches to predicting absorption in older population, what we should know and why. And two other ESRs from our consortium are going to present their research project on this, Donia Robbins and Cleo Dimnester, and the date, and please save the date, is Tuesday, 16th of April, same time. I would like to ask you to complete the survey, as Professor Epos pointed out, because you will help us to improve our webinars, and hope to see you in our next webinar on April. Thank you, and bye-bye.